From the JAMA Network, this is JAMA Neurology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Neurology.
Hello to our listeners from around the world. I'm Dr. Cynthia Armand, Associate Professor of Neurology at the Albert Einstein College of Medicine, Montefiore Medical Center, and JAMA Neurology web editor. Joining us today on the podcast is Dr. Susan Landau. Dr. Landau is a research neuroscientist at the Neuroscience Department of the University of California, Berkeley. She focuses on brain changes associated with healthy aging and Alzheimer's disease. She is the principal investigator of the Pointer Imaging Study, the topic of our discussion today. The Pointer Study stands for the U.S. Study to Protect Brain Health Through Lifestyle Intervention to Reduce Risk. And what Dr. Landau and her colleagues are looking into are brain imaging biomarkers and cognitive outcomes as they are affected by multi-domain lifestyle interventions. Dr. Landau, thank you for joining us at the podcast.
Thank you so much for having me. It's really a pleasure.
Can you tell me about the inspiration behind this study? And I want to start off by saying I'm very excited by this work. As a neurologist, we have a lot of patients who are concerned about their memory and their cognition and what happens as they age. And what can I do, doctor? What can I do so that I can halt aging in any way possible, especially my cognition and helping with my memory? And I just want to hear more about this.
Yes, so there really is a lot of public interest in how people can reduce their risk of developing dementia through lifestyle practices. And the ones that you hear about the most are diet, physical exercise, cognitive exercise like brain games. So the goal of the overall US Pointer trial was to do a really large and rigorous and hopefully definitive study to try to understand whether making changes in lifestyle across multiple areas of lifestyle practices, so multiple domains, physical and cognitive activity, diet, social engagement, health risk monitoring, whether making those changes could improve cognitive function. So this study was carried out over 2 years. And the results of the main trial were reported in the Journal of the American Medical Association last year. There was a cognitive benefit that was established for people who engaged in these lifestyle changes over 2 years, compared with people who did a less rigorous degree of these changes. So it's important to understand that in this particular trial, there really was no placebo group. There were two groups that were both told about the potential helpfulness of these lifestyle changes. And one group was asked to participate in these lifestyle changes at a much more rigorous level than the other group. So the group that had the rigorous, high-intensity changes was called the structured intervention group. And the other group was called the self-guided group. That group involved much less monitoring and frequent engagement or assessment of the degree of their engagement. So the outcome of the trial was really exciting. And as I said, it was reported in JAMA last summer. The structured intervention group had greater cognitive benefits over the 2-year trial period than the self-guided group. And so this was really seen as a very exciting development in our understanding of how lifestyle changes can improve cognition and potentially the risk of cognitive decline and dementia in older adults.
These two groups that you described from the original Pointer trial actually remind me of what happens when individuals have a goal and they may have a trainer or someone who guides them through certain interventions, and then other people's who don't and they do it on their own. And I think there's something to be said about frequent assessments, goal-driven, things of that sort in this capacity. But for this study, you're looking at imaging changes, correct?
That's right. So the particular focus of the results that we're talking about today was a sub-study that was carried out in about half of the participants who enrolled in the main trial. And the goal of this imaging study was to try to understand what brain mechanisms might underlie the cognitive benefits that were observed in the main trial in response to the structured lifestyle intervention.
So you were interested particularly in different markers of neurodegenerative disease as it relates to cognition, correct? So can you tell us about the primary imaging outcomes that you were looking at?
We enrolled and had imaging carried out in about over 900 participants who were also enrolled in the main trial. They had MRI scans at baseline, at 12 months, and at 24 months, which was the end of the trial. They also had amyloid and tau PET imaging at baseline and at 24 months at the end of the trial. And so we were really interested in quite a number of different measurements related to amyloid and tau pathology, both at baseline and how it changes over time. We're also interested in quite a number of structural and cerebrovascular measurements from the MRI scans. But our primary outcome measures for this imaging study were amyloid PET measured in a cortical summary region across the whole brain, tau PET measurements in entorhinal cortex and in a temporal composite region. Another primary outcome measure was white matter hyperintensities, and the last one was hippocampal volumes.
Now, did you have any other primary outcomes that you were looking into in terms of cognitively what you were following?
So the imaging measurements were the PET and MRI measurements that I mentioned, but we were also looking at changes in a global composite cognitive measurement over the course of the intervention period. And then we looked at three specific cognitive domains: processing speed, executive function, and memory function. And those cognitive measurements, the three specific domains and the overall global composite measure, were all the same as the ones used in the main trial as the primary outcome cognitive measurements in that trial.
Nice. And can you tell our listeners again, specifically, because this comes up in terms of what exactly is structured about the interventions? What exactly were the individuals doing? What did that look like? I know you mentioned cognitive activities, physical activity. What else were involved in those particular exercises?
The implementation of the structured intervention was an incredibly intricate aspect of this trial. And I should say that was developed based on the finger trial, which was another multi-domain lifestyle intervention study that was carried out in Finland. And that was a successful trial that also showed a cognitive benefit following practice of multiple cognitive domains. And so the way this concept of multiple cognitive domains over this trial period, the way this was implemented in the US Pointer trial was across 5 different locations in the US. Each of those sites had teams of site staff members. There were site navigators and site interventionists who were involved in meeting with the participants and making sure that they had all of the support that they needed to engage in all the different aspects of the interventions. It's kind of difficult to briefly summarize the implementation of the interventions because it was so elaborate. But for example, the physical exercise component, which obviously was a very important part of the intervention because it's one of the lifestyle changes that we think about the most and that there's the most evidence for in terms of both brain changes and cognitive changes. The participants in the structured intervention had a Fitbit that allowed the participants and the study staff to monitor how much exercise they were engaging in. So each component of the intervention has its own adherence measures to determine how much participants were engaging in that component of the intervention. I'll also say that there was a big social component to both intervention arms, but particularly the structured intervention arm, because participants within a particular site were actually assigned to teams and they met with that team and a site intervention group weekly for the entire intervention period. So for participants in the structured intervention arm, they had 38 team meetings with their group over the course of the trial. In the self-guided group, there were also team meetings, but it was a much smaller number. They only had 6 throughout the entire trial.
Mm. And that's a big difference. Thank you so much for explaining that because it really pinpoints the rigor with which these interventions were performed, especially in the structured group as compared to the self-guided. And that has implications for future studies and what we can tell our patients at the chairside and bedside, for example.
I agree with you. And the real world implementation of the lifestyle interventions was something that was very much in mind with the team that designed them. Making sure that, yes, it was very demanding and intensive intervention, but it was designed so that in the event that the trial was successful, which it was, would it be possible to implement on a nationwide scale so that clinicians could actually formally prescribe or recommend this intervention to patients who are interested in making those changes and participants who fit the at-risk profile of the participants enrolled in the trial?
Yeah, I really like that setup. Let's talk about the results.
Okay, so we had two major goals in the imaging study. The first aim of the imaging study was to look at whether there were longitudinal beneficial changes in our primary imaging outcomes, amyloid and tau PET, hippocampal volume, and white matter hyperintensities. And we did not see 2-year changes that were different between the intervention groups, meaning that all of the participants showed a similar amount of change in those imaging outcomes across both intervention groups. We really saw changes in those imaging outcomes that were expected for the characteristics of these participants aged 60 to 80. We saw subtle increases at the group level. For both intervention groups, we saw subtle increases in amyloid and tau, and we also saw some increases in white matter hyperintensities and decreases in hippocampal volume. So all of those were the kinds of changes we would expect to see in a group of older at-risk participants, but we didn't see differences in the amount of those changes between the two intervention groups. Our second main goal of the imaging study was to look at whether there were particular imaging-based characteristics or profiles that predispose people to do better cognitively if they were in the structured intervention group. And here we did find some really interesting profiles that predicted better cognitive benefit. So the first one was that we found that people who had lower hippocampal volumes at baseline were likely to benefit more cognitively from the structured intervention compared to people who had low hippocampal volumes at baseline and who were in the self-guided, the less rigorous intervention group. So this finding really pointed to this question of, are there particular types of people, are there particular characteristics that predict a greater benefit, a disproportionate benefit cognitively from lifestyle changes? And are finding that people who had lower hippocampal volume, people who may have already experienced some hippocampal atrophy, our finding was that those people did better cognitively over time. And in some ways, it parallels a finding reported in the main trial, which is that people who started off doing more poorly cognitively at baseline they actually had a disproportionate benefit from the structured intervention longitudinally. So the parallel finding there is that there seems to be this kind of at-risk profile performing more poorly at baseline, having lower hippocampal volume that allowed people to benefit more cognitively from the more rigorous lifestyle change group. We had another finding that kind of fits into this pattern of at-risk profiles benefiting more from the intervention, and that was that people who accumulated more tau over the course of the trial did disproportionately better as well cognitively if they were in the structured intervention. So in other words, the lifestyle intervention didn't reduce the amount of tau that people accumulated over 24 months, but those that were accumulating more tau, if they were in the structured intervention group, they did better cognitively than if they weren't in that group, than if they were in the self-guided group. So again, it points to this pattern where people who were in these at-risk groups based on their tau accumulation, based on where they started on hippocampal volume, and based on where they started cognitively, they were in a position to do better in terms of their cognitive benefits from the structured intervention group.
That is remarkable, especially for early intervention and guidance in terms of treatments and types of treatments, how we tell our patients to live their lives and move forward and actually add on to potential therapies that we may be prescribing.
I agree. And I wanted to add too that in terms of trying to determine which kinds of patients could benefit the most from lifestyle changes, because that's really what this kind of subgroup analysis that we did, looking for certain characteristics, that's what it was aimed to do. There's another finding we had that I think speaks to the clinical implementation of this intervention, and that is that the amyloid status that individuals had at baseline was not predictive of the degree of cognitive benefit that they experienced from the structured intervention, meaning that individuals who were amyloid positive at baseline and therefore on the pathway to Alzheimer's disease, even though most of the participants in this sample were cognitively unimpaired, those who were amyloid positive and on the Alzheimer's pathway didn't differ in terms of the cognitive benefit they could experience from the lifestyle intervention compared to those who were amyloid negative at baseline. So this finding overall really suggests that the cognitive benefits that people can experience from lifestyle changes are not specific to those who are on the Alzheimer's pathway or not on the Alzheimer's pathway. We are still looking into this finding to determine whether there are any lifestyle-related changes that we see either in the other imaging assessments that we did or cognitively. We're still looking to see whether there are any kind of Alzheimer's-specific effects that we see potentially through longer follow-up of the same participant group.
That lack of difference in intervention responses is really remarkable. And I wonder, we followed these patients over 2 years as part of the structure of the Pointer trial. Do you expect to have longer term data and even more improved differences in the results that we've seen here thus far?
The longer term follow-up of the Pointer participants is a really important aspect of this trial. It may be that the changes that we saw both cognitively and on the imaging outcomes were just the tip of the iceberg, since it's likely that both brain changes and cognitive changes happen slowly in response to the types of physiological changes that you might expect in response to those lifestyle modifications. So in order to really understand the longer term effects of the intervention, there's an extension study underway. The main trial is following participants cognitively for 4 years following their completion of the interventions. And the imaging study has an extension phase underway as well. We have already acquired 2-year post-intervention follow-up data in over 200 participants. So really the goal is to understand whether there might be longer-term changes, both cognitively and in the imaging measures, that allow us to detect this longer time course that we would expect to see. And I'll just mention that although we didn't see longitudinal changes in our primary imaging outcomes after 2 years, it may be because we know that, especially in a relatively unimpaired population, we don't expect to see really rapid changes in amyloid and tau, um, or in terms of white matter hyperintensities or atrophy. We don't expect to see major changes over 2 years, but we're really interested in looking at this longer time course. It'll be 6 years total, the 2-year intervention phase and the 4-year extension phase. We're really looking at that longer time course in this extension period.
Dr. Landau, I want to thank you so much for joining us today. And myself, and I'm sure all of our listeners, are looking forward to your future research in this arena.
Thank you so much. I really enjoyed getting to talk with you. And I also want to acknowledge Tessa Harrison, who is the lead author of this paper, the U.S. Pointer Study Group, and the Pointer Imaging Investigator Team for all of their contributions. Thank you.
I'm Dr. Cynthia Armand, and I've been speaking with Dr. Susan Landau about the Pointer Imaging Ancillary study. You can find the link to the article in this episode's description. This episode was produced by Daniel Musisi at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. Thank you for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.